Well, hello, and welcome to the Behind the Lab series. If you can just give us your name and what university you are attached to.
Yeah, my name is Nathan Winn. I did my PhD at the University of Missouri, and now I'm an assistant professor at Vanderbilt University Medical Center. And so I study metabolism and obesity. I'm interested in how obesity can amplify certain diseases like cardiovascular disease, diabetes, and fatty liver disease. We're interested in sort of mechanisms related to those aspects.
Okay, so as we get started, my first question is for a center director or faculty member, what notable work have you done while being a part of the MMPC?
So basically, we have one of the key aspects that my lab uses is a surgical technique in which we implant carotid artery and venous catheters. So that would be putting in catheters into a major artery in our neck as well as into the vein, and this is in rodent models. And this gives us access to the bloodstream of the animal. And the advantage of this is now we can study an animal in its normal physiologic state. And so we have multiple procedures that use nutrient tracers, so we can use isotopes, natural isotopes, that can reflect the metabolism of the animal. And we can manipulate those and understand how mechanisms of glucose, how it's regulated in the liver and the muscle and various other tissues and organs, including lipids as well. So it gives us a unique opportunity to study the mouse in its normal environment. And that's a huge advantage because in the past, we weren't able to do that. We had to put mice under anesthesia or study them in a non-physiological state. And so the major advantage, at least with regard to this technique, is that we can now do this in an animal that's unstressed and it gets better reflection of metabolism. And so these techniques, which is often simplified as an insulin clamp, has been modified over the years to improve the efficiency, the expense of doing these type of procedures. And so it's been really advantageous to my group because I'm interested in whole body metabolism and how that's influencing various organs in these disease states.
This is so awesome. This, I believe, also kind of ties into what my next question is. Like, with this being, I guess, a new technique that you guys have used, would you say that this is like a medical or research advancement that you guys that you have been a part of since being a part of the MMPC and being at Vanderbilt?
So the insulin clamp has been around for quite some time, and it was first established in humans. And so this is actually a technique that we have brought down to the rodent models, particularly in the mid 2000s when mouse genetics was gaining traction. And so we could manipulate genes in the animal to then study specific diseases and mechanisms. And so at the time, before this technique was modified to the mouse, we didn't really have the opportunity to study the in vivo condition because the animals were just too small, right? And so we had to rely on human data or large animal models in order to make inferences to human disease. But the big jump that was made when the MMPC was created was that now we have an opportunity to study specific genetics in a small animal model that still reflects the metabolism that typically occurs in humans. And so the insulin clamp has been established in the MMPC since its inception in the early or mid 2000s. But we've modified the technique to use different tracer technologies, and we have really skilled investigators here at Vanderbilt, including people like Jamie Young's group, who has used mathematical modeling technique to really expand the repertoire of information that we get from this clamp procedure. So the surgical technique that I described to you has been firmly established for 20 years now. But the information that we can extract from that is what has been modified over the years to really allow us to make more in-depth inferences and expand our information to multiple tissues that we may not have been able to interrogate.
Now, as a faculty member at Vanderbilt, have you been able to utilize the services that MMPC as a whole consortium offers?
Yeah, absolutely. And so
Okay.
the biggest one of the biggest advantages of the way in which the MMPC is run and works is access to the staff and access to the leadership. And so this is critical because, we as single investigators, when we're approaching an experimental design, oftentimes, of course, you have your colleagues that you can run ideas off of. You, of course, have the literature that you can go reference. But having a core group of faculty and staff that have such experience in these areas of running this type of core, you really have the unique aspect of sitting down with them and figuring out, okay, this is my question. This is what I want to get from this experiment. How can we design a complex in vivo experiment, at least in my case, to answer this question and answer this question in a rigorous manner? And so that is probably one of the most notable resources available is literally just the interactions with the staff and the faculty and other researchers on campus and abroad that have input into the way that you're designing and conducting the experiment. And that's unrivaled, I think, at most core facilities.
How long have you been affiliated with the MMPC, or how long have you been working at Vanderbilt to have been able to work on the specific project that you're working on and also see all of the collaboration between the individuals who have that experience and what you're doing?
Yeah, so as I mentioned earlier, I did my PhD at the University of Missouri. And so I came to Vanderbilt in 2018, where I started my postdoc. And my postdoc was with Dr. Alyssa Hasty, who was a former faculty member at Vanderbilt, who was a frequent user of the MMPC. And I was co-mentored in my later years of postdoc by our dear late Dr. Wasserman, who started the MMPC. So I had a outstanding experience with learning what the MMPC actually was, and what the capabilities of it was from the inception of my time at Vanderbilt, which started in 2018. So from the get-go, I started using the facilities when I was in Dr. Hasty's lab, and that included things like the body composition analyses. We have machines that allow us to measure an animal's energy expenditure. And so those were critical pieces of equipment at the time, because I was studying the effects of what you could consider yo-yo dieting in mouse models, right? And so those were key aspects of the research at the time. And then that grew, over the past almost 10 years now. So I've been, and then as an official component of the MMPC has been about the last year or two. Yeah.
Since working on your specific project, have you been able to publish any papers, or have you had any significant research milestones that you've hit so far?
Yeah, so I think I should have counted before I got on the call. I think we have, somewhere in the neighborhood of 12 publications, I think, over the past 5 years with the MMPC resources. And there have been a couple that have, you always go into a study with a hypothesis and what you think is going to happen. Mm-hmm. Oftentimes we end up being wrong, right? Okay. But some of the studies that we've done through the MMPC has really steered us in different directions that we did not necessarily think was going to happen, and they've been very fruitful. So as an example, I mentioned earlier that I study obesity, and one consequence of obesity is insulin resistance. And so insulin resistance, in simple terms, is the inability to respond to the hormone insulin. Mm-hmm. And so after we eat a meal, insulin rises in the blood, and one of its main jobs is to clear glucose from the blood so we don't have a high glucose levels. So we had an experiment in which we were doing these yo-yo dieting studies, and yo-yo dieting poses a unique metabolic challenge to both humans, and it's reflected in animal models. And it actually can potentially worsen insulin resistance. That was our hypothesis. And so we went in and we used the MMPC, we did some clamp studies, and we actually found that the mice were not as insulin resistant as we thought they would be. And it turned out that their pancreas was becoming, they had basically pancreatic fatigue, meaning that they weren't able to secrete as much insulin to then control their blood sugar. So that by using this MMPC related in vivo technology, I was able to shift my studies from mainly focusing at the time on muscle and liver to the pancreas, and developed an entire grant on that project that we're still continuing now. So if I had not used an approach that allowed me to survey different organs simultaneously, I would have missed that the pancreas was being or was not responding appropriately. So that's one example of how this using a robust technique that gives you a lot of information can open up doors that you didn't necessarily think was going to be there.